Hi everyone. Today we're going to be solving Cambridge International AS and A-Level Biology 9700 paper 52, February March 2024. Onions are vegetables that are harvested from the plant Allium cepa. In many countries, small onions are used to make pickled onions. Pickled onions can be stored for a long time because the growth of microorganism is prevented. Pickled onions are made using this method. Remove the dry outer layer from the small onions. Place the onions in sodium chloride solution for several hours. Remove the sodium chloride solution and place the onions in a solution of ethanoic acid vinegar. Figure 1.1 shows a container of pickled onions and some fresh onions that have not been pickled. So some students decided to investigate how placing small onions in different concentration of sodium chloride solution affect the mass of the onions. Identify the independent variable in this investigation. Independent variable is always the variable that we can change. So different concentration of sodium chloride solution. The students were given a 20% stock solution of sodium chloride. The students decided to use the stock solution to make sodium chloride solutions with concentrations of 0.1%, 0.0%, 1%, 5%, 10%, 15%, and 20%. The student made 140 cm³ of this solution in separate beakers. Complete table 1.1 to show how 140 cm³ of these solutions could be made by proportional dilution of 20% stock solution of sodium chloride. So, in order to make the diluted solution, we'll have to add water. So volume of water slash cm³ that we will be adding. In each cases, we are going to prepare 20%. We are going to prepare 140 cm³ solution. So the formula that we can use in this process is V1 times C1 is equals to V2 times C2. For the first one, let's do this. So for example, for concentration of 1.0. For concentration of 0, we can have 0 cm³ of sodium chloride and the 140 cm³ will be taken from the water. So that's a no-brainer. However, for 1% sodium chloride solution, we have to make the solution in this way. The V2 is 140, which is fixed, and the concentration that we want to make is 1. The V1 we do not know, and the concentration of the V1 is 20. So if we take V1 and 140 divided by 20 will give us 7. So we will require 7 cm³ of this, and the remainder will be water, so which will be 133. The volume of water needs to be written as distilled water. So the same math we will apply. In order to do for 5%, it will be 140 times 5 divided by The students carried out this procedure. The dry outer layer was removed from 30 small onions. 5 of the small onions were placed into each of the 6 beakers containing sodium chloride solution prepared by the students. The onions were left in the sodium chloride solution for 2 hours. The mean percentage change in mass of 5 small onions in each beaker was calculated. The students then repeated the whole procedure using a new set of 30 small onions. This time, the onions were left in the sodium chloride solution for 48 hours. Figure 1.2 shows the results of the investigation. So when the mean percentage change in mass is 0 at that particular point, it would be the concentration of similar to that of the onion. Whatever the onion's concentration is, the water potential are same at that point, which is why the mean percentage change in mass is 0. Explain why the student calculate the percentage change in mass of the onions. This is very simple because different onions will have different mass. So percentage change in mass allows valid comparison. So we should say allows valid comparison between onions in different sodium chloride solution because starting masses are not constant. So we can see after 48 hours, the percentage change in mass at the concentration 3.2 is 0 and 4.3 it's 0 but only experiment which is done for 2 hours only. This is for the 48 hours. One of the students concluded that the water potential of the onion cell is the same as the water potential of a 4.2% sodium chloride solution with the reference to the information provided, including the results shown in figure 1.2. Such is the reason why this conclusion should not be accepted. So this experiment was done in 2 formats. One was after 2 hours conclusion and one was after 48 hours there was a conclusion. So in the experiment that was done 48 hours, we see that at 3.2%, we can see there is 0% change. And in the experiment that was done for only 2 hours, we can see at 4.3%, there is 0% change. So we can definitely say, all right, the conclusion is only supported for onions that are left for 2 hours. So the answer can be written as follows. The conclusion is only supported for the onions that is left for 2 hours. The intercept of the x-axis or the 0% change when left for 48 hours is 3.2% sodium chloride. So this produces a significant experimental error. All right. One of the reasons that we can say that this particular test is not a representative of the original of how a test should be, all right, or we cannot take the conclusion as accepted. That's because we did not do any intermediate concentrations of sodium chloride that we tested on that particular onion. So there was not enough information there, all right, and not all onion cells have the same water potential as well. The turnip Brassica rapa is not the turnip Brassica rapa is a root vegetable. The turnips are grown in many parts of the world. Figure 1.3 shows freshly harvested turnips with the leaves still attached. A student decided to investigate the effect of temperature on the rate of osmosis in turnips. In an initial test, the student removed the outer layer from a turnip and cut the turnip into small blocks. The student placed one of the turnip blocks into a beaker of distilled water, as shown in figure 1.4. Water entered the block by osmosis. So we can see the turnip block. Water is entering the block by osmosis because the water potential inside the block is lower than the distilled water for sure. So water is moving inside the block. The mass of the block will increase after a certain period of time. The student was provided with additional turnips and standard laboratory apparatus. Describe a method that the student could use to investigate the effect of increasing temperature over a range of from 10 degrees Celsius to 50 degrees Celsius on the rate of osmosis in the turnips. Do not include the risk assessment or how to calculate the rate of osmosis from the results. So we are not to include any risk assessment and how to calculate the osmosis. Your method should be set out in a logical and detailed enough to allow another person to follow it. So in order to do a experiment that has a range of temperatures, we must have at least 5 stated temperatures, all right, with a suitable method to maintain the temperature. So we should have like a water bath to maintain the temperature. And to make the experiment fair, we should have same variety and age of turnips. Along with that, whenever we're going to cut the turnip block, it's going to be the same dimensions as well, all right, so that the surface area exposed to the water is the same. To ensure that the surface area of that cut block is same, we can use a 1 centimeter by 1 centimeter square cutting block, which can ensure that we are going to have 1 cm cube of block each time. For each temperature, we will place the turnip block in beaker, and at least 5 turnip blocks should be placed for each solution. For each temperature, we should place at least 5 turnip block in that temperature water. And then we should test for each temperature and calculate the average. How we are going to do it? We will also take the initial mass and the final mass of the turnip blocks, and we will dab it with a paper towel so that we can remove the excess water from the turnip block before measuring it. Complete the sketch graph shown in figure 1.5 to predict the effect of increasing the temperature over a range of 10 degrees Celsius to 50 degrees Celsius on the rate of osmosis of turnips. Include axis labels with units in your answer. So on the x-axis we can write temperature because it is our independent variable, and on the y-axis we can write rate of osmosis. Now as the temperature increases, rate of osmosis should increase. Suggest one hazard of your method in C1, the risk associated with the hazard, and the precaution that would need to be taken. So one of the hazard that we can definitely mention is that the use of knife or blade. So there is a risk of injury, and precaution would be cut away from the hand, cut down onto a board. The other one that we can mention is turnip itself is a hazard, so it can be removed. The risk is that it can cause allergy, so it's an irritant. So wear gloves. Drosophila melanogaster is a small fruit fly that is often used in research on genetics. While fruit flies normally have dark red eyes due to the presence of brown pigment homochrome and a bright red pigment dorsopterin. Figure 2.1 shows an adult female fruit fly and an adult male fruit fly. So we can see the adult female and the male fruit fly has a difference in pointed abdomen and rounded abdomen. A biologist carried out an investigation to determine the roles of two genes that are involved in the determination of eye color in adult fruit flies. Both genes have two alleles. Earlier research by other scientists suggested that the synthesis of brown pigment homochrome depends on the gene B/b. The synthesis of the bright red pigment dorsopterin depends on the gene R/r. The biologist obtained the parent fruit flies shown in table 2.1. Parent fruit flies female B smaller produced brown eyes, and the male b R, bright red eyes. The biologist carried out the procedure shown in figure 2.2 to cross the parent fruit flies and obtain the first generation offspring. Specimen tube with agar jelly containing nutrients and water. Some agar jelly containing nutrients and water was placed in the bottom of several specimen tubes. On day 1, 4 adult female fruit flies with brown eyes and 4 adult male fruit flies with bright red eyes were placed in each specimen tube. A piece of cotton wool was placed in the top of each specimen tube. The specimen tubes were left in a warm room for several days. During this time, the parent fruit flies reproduced and laid fertilized egg on the agar jelly. The fertilized egg hatched into larvae that feed on the agar jelly. On day 7, the parent fruit flies were removed from each specimen tube. By the day 12, the first generation of adult fruit flies had developed from the larvae and were ready for crossing to produce the second generation. Suggest a reason why the biologists removed the parent fruit flies from the specimen tube on day 7. It could be because the parent fruit flies might eat the larva. However, the answer is to prevent the fruit flies from reproducing or mating with the first generation offspring. The biologists wanted to cross the first generation of fruit flies with one another to produce the second generation of fruit flies. The first generation of adult fruit flies in the specimen tube on day 12 were a mixture of female and males and had not yet mated. The biologists crossed the first generation of fruit flies with another by preparing fresh specimen tubes in which to produce the second generation of fruit flies using a chemical to anesthetize the first generation of fruit flies so that they were temporarily unable to move, separating the adult female fruit flies and adult male fruit flies. Placing 4 adult female fruit flies and 4 adult male fruit flies into each of the fresh specimen tubes suggests a method that the biologists could use to separate anesthetized adult female fruit flies and anesthetize adult male fruit flies and place 4 of each into a fresh specimen tube. Now the answer we can write down. To separate the male and female fruit flies, the scientist can use a hand lens to find whether they are male or female. And to place the fruit flies, the scientist can use a paintbrush. The biologists expected the cross to result in a phenotypic offspring ratio 9 to 3 to 3 to 1 in the second generation. Table 2.2 shows the results of this cross. We can see the dark red eyes, expected phenotypic ratio 9 and observed number 691, and then the other ratios are provided with their observed number. The biologists used the chi-square test to compare the observed and expected result for this cross. State the null hypothesis for the chi-square test. So we will say there is no difference between the observed and the expected result. To calculate the equation for chi-squared is equals to sum of observed minus expected whole squared divided by the expected. According to the ratio 9 to 3 to 3 to 1, the expected for the dark red eyes are 702. So the observed minus the expected. So after doing the whole maths, the chi-squared value is 3.869, which means the degree of freedom is 3 because we have 4 observable class and our exam probability is 0.5. So it has a value of 7.815. Now our chi-squared value is 3.869, which is less than 7.815. So the critical value The biologists compared the calculated value of chi-squared to the critical value at different probability values shown in table 2.4. Using the table 2.4 and the calculated value of chi-squared in C2, explain whether the null hypothesis should be accepted or rejected. So let's see the critical value first. The critical value is 7.815 and our chi-squared is less than 7.815. So the null hypothesis is accepted. Whenever chi-square value is less than the critical value, the null hypothesis will be accepted. More recent research has shown that gene capital B/b and the gene capital R/small r code for polypeptides in carrier proteins. These carrier proteins are found in organelle membranes of the pigment cells of the eyes of adult fruit flies. The gene capital B/small b codes for a polypeptide in the tryptophan carrier protein. Tryptophan is an amino acid. The gene capital R/small r codes for a polypeptide in the guanine carrier protein. Figure 2.3 shows how the dark red eye color of wild fruit flies is produced in organelles in pigment cells of the eyes. Tryptophan carrier protein, we can see tryptophan in this then converted into homochrome, which is the brown pigment, and guanine, which is dorsopterin, the bright red pigment. The biologists decided to analyze all the pigment present in the eyes of the second generation of fruit flies. The biologists started by extracting eye pigment from the adult fruit flies with dark red eyes and from the adult fruit flies with white eyes. The biologists then added a small volume of each liquid extract to chromatography paper and separated the pigment present by chromatography. Figure 2.4 shows that chromatography paper at the end of the procedure, when viewed using visible light and ultraviolet light, pigment 1 and 3 were visible only when viewed under ultraviolet light. Under visible light, pigment 2 was yellow and pigment 4 was red. So in the next one, we can see the chromatogram. We can see the position where liquid extract was added from fruit flies with dark red eyes. And we can also see the position where real liquid extract was added from fruit flies with white eyes. The biologists calculated RF value for each pigment on the chromatography paper. The biologists used the RF values to confirm the identity of the pigments using a published source. The formula for the calculation of RF is distance moved by the pigment divided by distance from origin to solvent front. When measuring the distance moved by the pigment, the distance to the center of the pigment should be measured. Figure 2.4 we have to use to calculate the RF value for pigment 1. So we will have to measure the distance from the pencil line up until the solvent front, and then we will have to measure the distance from the pencil line up until the middle of the pigment 1. The RF value by measuring the distance is calculated to be 0.73. State the conclusion that can be made from the results of the fruit fly breeding experiment in C and from the chromatography results. So from the chromatogram, we can give the observable difference in this way. Fruit flies with dark red eyes have 4 pigments in their eyes. Fruit flies with white eyes have no pigment in their eyes. Pigment 4 is dorsopterin. And finally, about their genotype, we can say the fruit flies with white eyes have the genotype b, small r, homozygous recessive for both genes. Guys, that's all for this particular question paper. Thank you for watching this particular video. Guys, question paper videos like this will be uploaded regularly. So subscribe to the channel so that you can be notified when a video like this is uploaded. And best of luck for your exam, guys. See you in the next video, guys. Bye-bye.